Now see for meiosis. Meiosis is divided into meiosis 1, meiosis 2. And meiosis 1 is again divided into prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. Prophase 1 is divided again into 5: leptotene, zygotene, pectine, diplotene, and dikinesis. Now I'll teach you my way first, then you understand the concept carefully, then I'll show you NCERT. Now see beta, in the leptotene stage, I'm drawing this one circle as a nuclear membrane, but you know nuclear membrane is double. But you understand, I'm drawing it one line. I'm taking a cell, and if you see here right now, this sister chromatid is not visible. This is one chromosome. This is second chromosome. Okay. I'll use this color and this color. So I've taken a cell with 4 chromosomes, and if you see my diagram, ends are still attached to the nuclear envelope. So this is your leptotene stage. Chromosomes are attached to the end of nuclear envelope by the help of attachment plate. You call this stage as bouquet stage also. Bouquet stage. Now the cell is going for the next stage, that is known as zygotene. After leptotene, cell is entering into the zygotene. If you see beta, these two are homologous chromosome. So pairing will start in the homologous chromosome. This is homologous chromosome. Sister chromatid is there, but still not clearly visible. Now here the pairing will start. To make the pair, you are getting a ladder-like structure in between them. This is known as synaptonymal complex. So zygotene stage is the one in which pairing will occur between homologous chromosomes. Now, what are homologous chromosomes? Homologous chromosomes are those in which sequence of the gene, position of the gene, size of the chromosome, everything is same. Allele can be different, but position of the gene and sequence of the gene will be same. Size is also same. So first here the pairing will start. When the pairing will start, we will say bivalent are formed. Bivalent are made. Bivalent means I will say synapses are made. In my diagram, I have taken how many chromosomes? Total 4. But how many bivalent? 2. So see beta, 1 bivalent means 2 chromosomes. So 1 bivalent means 2 chromosomes. And in between them, what I am finding? I am finding synaptonymal complex. I am finding synaptonymal complex. This much I hope so is clear to you. Clear? Now see, next is your pectine stage. I'm just trying to draw it big so that I can explain it properly. Now see the chromosome. Now I think so in each bivalent, I'm able to see 4 chromatid clearly. And I'm finding overlapping is going on. So this Pacatine stage is the one in which I'm able to see tetrad, all the 4 chromatid clearly. It means my numerical question, if I want, I can ask you how one bivalent has how many tetrad. You will say it is one bivalent is having 1 tetrad. One bivalent is having how many chromosome? You will say 2 chromosome. 2 chromosome. One bivalent is having how many chromatid? If I ask you how many chromatid, then you will say 4 chromatid. 4 chromatid. In this stage, what is going on? Crossing over is occurring. Between whom? Between non-sister chromatid. Because beta, orange color 2 are sister, green color 2 are sister. But right now I'm doing crossing over between non-sister chromatid of homologous chromosome. So when crossing over is occurring, means exchange is going on. So here when the exchange is going on, so I think so I have to exchange the genetic material from green color to orange color. So I think so I want the set of an enzyme so that the gene can break from here and go. So I will say recombinase enzyme is present in the recombination nodule. Recombinase is present in recombination nodule. So I will find recombination nodule are made in which crossing over is occurring, and they are having the set of an enzyme known as recombinase, which is nothing, you can say your restriction enzymes which are going to cut, and after the exchange sealing will be done. So for sealing you want ligase enzyme. So this is very, very important stage in the meiosis, which is responsible for creating the variation. Now next stage, diplotin stage. Check my diagram carefully. See here, exchange is done. It will be overlapped, but I can't draw it here because otherwise you'll get confused. So I'm drawing it separately. Still overlapping is there, okay, beta, but I have drawn separately just to make you understand. Now see here, beta, this is your synaptonymal complex which was present. I have to now remove it. So in diplotin stage, synaptonymal complex dissolve. In diplotin stage, synaptonymal complex dissolve. Very important, first of all. First is synaptonymal complex dissolve. When the synaptonymal complex dissolve, what will happen? Now homologous pair want to separate, but they can't separate. They are united at the point where the crossing over is done. As a result, you are getting X-shaped structure. And this structure is known as chiasmata. And this is the point which indicate where the crossing over is done. Point to indicate crossing over is occurred here. Diplotin is characterized by the dissolution of synaptonymal complex and formation of chiasmata. Now, and diplotin is the longest stage in the primary oocyte in the female. If you know, in the human reproduction chapter we studied, the primary oocyte starts its meiosis 1 in the and prophase 1 reach to the diplotin stage and stop, and birth of the female child occur. It will start, it's enter into the dikinesis and complete at meiosis 1 at the puberty. So this is the longest phase. Now in dikinesis, what is going on? In dikinesis, nuclear membrane will completely disappear. Now your Chiasmata is trying to reach to the tip. Terminalization of chiasmata we have to show. So you have to just make it attached to the tip only. Just to the tip point, you have to make it attached. Because terminalization we have to show. Clear, beta? So this is your dikinesis in which terminalization of chiasmata is done. Where terminalization of chiasmata is done. Now 1 minute, beta. Now see, beta, we have studied prophase 1. It is subdivided into following 5 stages. First is leptotin. Chromosome gradually become visible under the light microscope. Compactation will continue. Only this much you have to remember for leptotin stage. Now go for the zygote. Second stage is zygote, second stage. During this stage, they start pairing. And the process is known as synapsis. Please remember, beta, this is spelling synapsis, and in the nervous system chapter, we study that is synapse. That is ending with SE, here SIS. And such paired chromosomes are known as homologous chromosomes. Electron micrograph indicates that the chromosome synapsis is accomplished by the formation of a synaptonymal complex. Okay, so synaptonymal complex is made between them. The complex formed by a pair of synapse homologous is known as bivalent also. Or you can say tetrad. However, these are more clearly visible at the next stage. That is why we say in the zygotine stage, we focus on the synaptonymal complex, we focus on synapsis, and we focus on bivalent. Because tetrad, we say it is more visible in the next stage. That is pachytene stage. Now, first 2 stages means leptotin and zygotine. The first 2 stages are short-lived. First 2 stages are short-lived. Pachytene is more long. It is taking more time. During this stage, 4 chromatids are clearly visible. That is why tetrad stage is more clearly seen in the pachytene stage. Here you will find recombination nodule. This is the site where crossing over is occurring, but it is occurring between whom? Between non-sister chromatid of homologous chromosome. What is crossing over? Crossing over is nothing. It is an exchange of the genetic material, which is occurring between them. Crossing over is also an enzyme-mediated process. I told you they want a set of an enzyme, restriction enzyme. After the exchange, they want to seal the ligase. So all these enzymes together in the set are known as recombinase. Crossing over lead to the recombination of a genetic material. Recombination between homologous chromosomes, so new combinations are coming. It is completed by the end of pachytene. So by the end of pachytene, the crossing over is completed. Diplotin stage. Next is 4th stage is diplotin stage. Dissolution of synaptonymal complex will be going on. And tendency of recombined homologous chromosome, they separate. They want to separate now from each other, but they are live at one point where they can't separate. So these accept at the site of crossing over, and these X-shaped structure are known as chiasmata. This is known as chiasmata. In oocyte of some vertebrates, I think so human best example, we can remember it. Diplotin can last for months or for years. For human, it lasts for years. Clear, beta? Dikinesis, terminalization of chiasmata. Obviously, this is the end of the prophase 1. So prophase 1 is end. It means nucleus, nuclear membrane, nucleolus, all things will disappear. Chromosomes have achieved the full condensation. Meiotic spindle is assembled. By the end of dikinesis, nucleolus disappear, nuclear membrane disappear, and now dikinesis means transition to metaphase. Again, statement question. Dikinesis represents transition to metaphase. Please remember this. Now let me explain you from diagram. Come here. Remember this diagram. We are taking how many chromosomes, beta? We are taking 4 chromosomes. Okay, we are taking 4 chromosomes. So now we are starting with the metaphase. In the metaphase 1, check my diagram and then tell me one thing. Let me draw it. Now see carefully, beta. This kinetochore and this kinetochore. Each chromosome, you know, it is having 2 kinetochore. And till now, if you see, chiasmata is maintained. Chiasmata is maintained. And see carefully, this is your spindle fiber. I'm drawing it. Check. I'm using only 1 kinetochore per chromosome. I'm not able to use 2 kinetochore. I'm able to use only 1 kinetochore per chromosome. And as a result, if you see, I'm making 2 metaphase plate. I'm not making equatorial plate. Please, I'm making 2 metaphase plate. Now for this imagination, I used to ask the student to remember one thing. Suppose I am hosting the birthday party of, let us assume I invited 23 small girls and 23 small boys. Okay, 23 small children. Age group is only 1 or 2 years old, not more than that. And I'm asking one girl has to hold the hand of one boy. And condition applied is this: if you will maintain the handhold, I'll give you pizza, and return gift will be Barbie doll to the girls and racing car to the boys. I think so no one will leave the hand. They will try to hold each other hand. So it means chiasmata is maintained. Now after 1 hour, I'm finding the children are hungry. So I'm asking them, please come in the middle of the room. So tell me one thing, I will find 1 row, I will find 2 rows. Logically tell me. I think so I will find 2 rows. I will not find 1 row. I will find 2 rows. 23 children in 1 row, 23 children in another row. And what I'm finding in the middle, I'm finding their hands in the middle. It means I'm finding chiasmata in the middle. The chiasmata hand is in the middle. Is it clear? And now this metaphase plate is metaphase 1 is very, very important. Understand independent assortment here. Now this arrangement, let us assume with a first pair where the girl is there on the left. Let me draw the diagram. Understand? One child here, girl is there. This side is boy. Second is also girl. This side is boy. Is it necessary कि here now again boy will come? Here girl can come, na? Anything possibility? We can change it. Anything possibility will be there. So when one set was aligning, they know we have to separate. We have to separate. Was it influencing this pair? No. It can come in this manner also, and it can come in this manner also. So same, imagine beta, when capital R and small r is separating, capital Y, small y is separating. You know very well they have to separate in the dihybrid cross. They can come in this manner also, and they can come in this manner also. Am I right? Any possibility? They know, this fellow know कि I have to separate. Same, this fellow also know I have to separate. But I think so orientation no one is controlling. That is why law of independent assortment says this only. Segregation of one pair is not affecting the another pair how to separate. As a result, beta, see this side pole you are getting capital RY gamete. Here you are getting small RY gamete. And this pole what you are getting capital R small y gamete. And here small y R capital Y gamete. So you are getting different types of combination just because of the orientation of homologous chromosome during metaphase 1. So this stage is very important to understand the law of independent assortment. one pair is not influencing another pair how to arrange. And till now, if you see chiasm meta is maintained. Till now, chiasm meta is maintained. Now, see the diagram. During anaphase 1, what is going, beta? Check. Homologous chromosomes are separating, sister chromatid are not separating. Homologous chromosome will separate, but sister chromatid will not separate. Understood, beta? It means both sister chromatids are reaching to the same pole. Both will reach to the same pole. Let me draw the diagram first, complete it, then I'll explain the diagram. And to study this better, you have to use this color only, then only the picture will become easy for you. This is your anaphase 1. Now at this phase, properly, chiasm meta is broken. At this phase, only properly chiasm meta is broken. Now the homologous pair are separating. Better now imagine, if the separation is occurring, I will say disjunction is occurring. But if separation does not occur, what will I say? Non-disjunction or not? You know one thing you have studied in the genetics chapter, Down syndrome. Down syndrome is because of the non-disjunction of homologous chromosome. Homologous chromosome fail to separate. You know one thing, just imagine we are studying the meiosis in the female ovary. This is going in the primary oocyte. And if suppose anaphase 1, 21st chromosome move to the same pole only. Both pair move to the same pole. And that becomes as a secondary oocyte. And that secondary oocyte later on become as a ovum. So this egg will be having 2 copies of 21 or not, beta. And this 2 copies of 21 will fertilize with the sperm. The child is born with the 3 copies of 21. We say child is suffering from Down syndrome. So anaphase 1 is the responsible for all these problems. We see chromosomal disorders is just because of anaphase 1, where the homologous chromosome failed to separate. Just to prevent such type of deformities in the children, we are doing the amniotic fluid test. If you remember in the reproductive health, amniotic fluid test, the test is known as amniocentesis, in which chromosomal abnormality is identified, and if suppose it is there, couples are suggested to go for MTP, which is legalized for few cases. But people started misusing to know the last chromosome. To know the last chromosome. Last chromosome, why? Because last chromosome in human determine the sex of the child. So anaphase 1 is clear. Now it will enter into the telophase 1. So in the telophase 1, I am directly drawing the 2 cells. I started a cell with how many chromosomes? Beta 4. Now I made 2 cells. Each is having only 2 chromosomes. So this is the phase where the actual reductional division has occurred. So in the telophase 1, I am finding I've made 2 cells, each cell with only 2 chromosomes. How many cells I started, beta? I started how many chromosomes? 4. 1 cell with 4 chromosomes. And I finally made 2 cells, each with how many chromosomes? Only 2 chromosomes. Now I have to separate sister chromatid. I have to separate sister chromatid. So this stage is also known as dyad of cell. This is known as dyad of cell. Now it will go for the short phase. That is, short phase is known as interkinesis. It is going for interkinesis. Interkinesis, why it is going? Reason is because this cell only got 1 centrosome. This cell also got only 1 centrosome. They want time to prepare second centrosome. So it means RNA formation and protein formation will be done. No DNA replication, please. Interkinase is a phase in which only RNA and protein synthesis will be done. DNA replication will not be done. Is it clear? Now I'm directly entering into the metaphase, because prophase nothing. Again, the nuclear membrane will disappear, all these things. So I hope you can understand that thing. Let me draw the diagram. This is your metaphase 2. This is your metaphase 2. Now in this diagram, tell me one thing. Till now, how many spindle apparatus I have made, beta? I think so I have made 3 spindle apparatus. Check carefully my diagram. One I made here, second and third. So one meiosis use how many spindle apparatus? 3. Again, your numerical question. One meiosis use how many spindle apparatus? 3 spindle apparatus. Now it is entering into the anaphase 2. So anaphase 2 is the phase where the sister chromatids are separating. Now sister chromatids are moving to the opposite pole. Sorry, space is less. Wait. Anaphase 2, where the sister chromatids are separating. And finally, you are going for the telophase 2. Understood better diagram? Everyone? So this is telophase 2, also known as tetrad of cell. Tetrad of cell. This is dyad of cell, this is tetrad of cell. Now you do one thing, I'll suggest you it. Now you analyze this entire meiosis properly. You analyze this entire meiosis properly. I think so picture will become very easy for you now. Very, very easy. Now let us see the NCERT beta. Bivalent chromosome aligned on the equatorial plate. Microtubule from the opposite pole attached to the kinetochore of homologous chromosome. Only one kinetochore is used. See beta, only one kinetochore is used. And you are getting this thing, 2 plates, you are making 2 plates. Anaphase 1, homologous chromosome separate. Sister chromatid remain attached. Is it clear? Now telophase 2, nuclear membrane, nucleolus will reappear, cytokinesis will occur. We are making dyad of cell. Dyad of cell. Chromosome do not undergo some dispersion, they do not reach. Stage between 2 meiosis is interkinesis, it is short-lived. Please remember, it is not interphase. Don't confuse with interphase, it is interkinesis. There is no replication of DNA. After that, it will go for prophase 2. Prophase 2 is just like mitosis. Meiosis 2 resemble normal mitosis, nuclear membrane will disappear. Metaphase 2, they will come and align at the equator. Microtubule from opposite pole get attached to the kinetochore of the sister chromatid. Earlier, they were attached to the kinetochore of homologous chromosome. Now they are attached to the kinetochore of the sister chromatid. In anaphase 1, what is going on? Splitting of the centromere will be done. Sister chromatid will be moved to the opposite pole by the shortening of which microtubule, which is attached to the kinetochore. This only I try to explain you while explaining mitosis. So this is the one continuation is going on. Check now each cell. Clear, beta? Telophase 2, in which they again enclosed by nuclear membrane and they are making tetrad of cell. Please remember, if they ask you tetrad stage, then it is Peckytine stage, but if they ask you tetrad of cell, then it is telophase 2. To make the 4 haploid. So how many chromosomes we started, beta? If you check here, we started a cell with 4 chromosomes. Clear na beta? In the diagram, we made 2 cells with 2 chromosomes. Again, each cell is dividing into 2, again with 2 chromosomes. Clear, beta? Summary is clear? Now see the numerical questions based on it. If suppose I ask you a question, कि one PMC, one colon mother cell has 20 picogram of DNA in G1 phase. How many DNA is present in the gamete? This is my question. How to solve? Do one thing. First of all, start this way. Draw 2 chromosomes in the G1 phase. In the S phase, do the doubling for them. In the G2 phase, obviously it will be doubled. This condition. Then prophase, metaphase will be this only. Anaphase, opposite pole. In the telophase, what will happen? You will have only 1 chromosome per cell. And in the telophase 2, you will have again 1 chromosome per cell. To do one thing, write down chromosome. You are starting with 2 chromosomes. It will be 2 till telophase, till anaphase. And finally, in the telophase 1, you will say 1 chromosome, and telophase 2, 1 chromosome. So this way. Clear, beta? Now DNA 2, here 2 is becoming 4, 4, here 4, and here 1. Now my question, if I'm asking 20 picogram of DNA in G1 phase, so G1 phase you write 20 here, and my question was how much in gamete? So put a question mark here. When 2 became 1, the 20 will become what? Simple. If you copy this presentation in your rough note, in the NEET exam paper also, I think so whatever question they ask you, you will not do mistake for the meiosis. Do this presentation on the side and start solving it. Now, one more question. I'm saying gamete has 5 picogram of DNA and 5 chromosomes. How many are present in megaspore mother cell in G2 phase? You have to tell me the DNA, you have to tell me the chromosome. To do one thing, again, work for this concept. Again, apply this concept. I'll first rub this area. What they have given us, they have given DNA 5 picogram, DNA 5 picogram, and chromosome number 5 they have given us. And they're asking G2. So G2, my question mark is here. They're asking me this content. 1 is 2, so here answer will be 10. Here 1 is 4, so here answer will be 20. Understood how to solve? Beta, understood? Now, one meiosis use how many spindle apparatus? 3 spindle apparatus. That I have shown you from the diagram. I think so. Topic is clear to you. Just go.